for doing the presentation. All right, the meeting has started. Peter basically has, since he left Williams and went to medical school at Tufts, has been involved in a lifetime of dealing with and studying infectious diseases. He received his MD from Tufts, did a residency at Yale, New Haven, and at Stanford Medical Center, and his pediatric infectious disease training at UCLA. He taught at UConn as a professor of pediatric infectious diseases, and moved back to Yale in 2008, where he's continuing his work. Peter's written over 2,000 peer-reviewed
200.
articles.
200. 200.
200, I'm sorry, I said 2,000, 200. Yeah.
I wish it were 2,000.
I wish it was 2,000, right, 200. And it's 45 book chapters and edited 2 medical books. He's well known in the field, and I don't know how many we just looked. I guess I'm the only one who's had Lyme's disease, but you've been studying several diseases that are borne by the ticks and have made a nice career of being able to go out to Block Island quite a bit. And that's a great place to be and do your work. So really take it away, Peter.
John, thanks so much for those kind words. And one of the advantages of my using this laptop is you can see my ceiling. In any event, so I did want to thank the seminar committee for inviting me to speak tonight. It's certainly an honor and a privilege, and I really appreciate it. And I especially appreciate Alan and John's help with getting the talk together. So I have carried out research principally on babesiosis, but also Lyme disease and other tick-borne diseases for the past 30 years. And I am going to talk a little bit about how I got involved with this research, but I'm going to start with a story. I call it the China babesiosis story. So after about 10 years ago, I got a call from the Chinese embassy in Washington, D.C., and they asked me if I would be willing to come to Beijing to evaluate a very VIP person with possible babesiosis. And they said they would, all expenses paid, but would I be willing to do that? And I said, yes, I would. And I said, when did you want me to travel there? And they said tomorrow. And so I sort of took a deep breath and said, okay. So the next morning, a large black SUV it comes into my driveway. Three fellows hop out. They all have trench coats on. They come to the front door, knock. My wife's standing next to me. She asked when I would be returning. And one of the fellows joked, said, "Are you worried he is never coming home?" He actually said that, words to that effect. And then everyone laughed. And I still was a bit anxious, but anyway, we get in the SUV, and I lived in Old Saybrook at the time, and we're traveling to JFK. On the way in New Haven, we
Peter, we lost your audio. Can't hear you.
We lost your audio. Yep. No, can you then on your audio down in the left-hand side or up at the top? There
okay,
you go.
can you hear
All
me?
right, you're back.
Okay.
I put him back.
What else can go wrong? Anyway, so anyhow, this guy pulls out a wad of dough to pay for this medicine. It's like the size of a toilet paper roll, and they're all hundreds, and he's just flipping these. Anyway, that was quite impressive. So we arrive in JFK and get on this China Airlines 747. I go up to first class where I've been instructed to go, and excuse me, I meet 3 other physicians who've also been called to evaluate this patient. I was the babesiosis person, but there were 3 infectious disease people, one from Mass General, one from Mount Sinai, and one from University of Texas in Houston. And we also learned that the person that we're going to be evaluating was a former premier of China. So that was interesting news. And anyway, we arrive in Beijing and we get off the plane before anyone else does, before any of the first class passengers get off. we were sort of whisked to these. There were four black Mercedes-Benz, with diplomatic flags on the front. We start speeding through Beijing. Cars, it's as if we were the police because cars pull over to the side as we're zipping by. We arrive at the hospital, at 45 minutes an hour or so, and very well appointed hospital, but we don't go to that hospital. There's a hospital behind the hospital, which is a military hospital. And so we get out of the car, and we climb a flight of stairs, open up this conference room doors, and there's a large conference room with, I'd say, 30 to 40 Chinese physicians. And the head of the group was a really nice guy, spoke perfect English, was a surgeon, and just coordinated we just started to evaluate. We hadn't gone to our hotel, just started the evaluation, which involved, reviewing all the past history, the present illness. We reviewed the lab results. We're not allowed to go in and see him that day. It was only the next day where we were allowed to see him. So, and the next day we walked into the room. We were told we could not do a physical exam. We couldn't lay hands on him. I think because they were concerned perhaps we'd have some pathogen on our hands and, that would negatively affect him. But he was a very interesting guy. He was sort of big booming voice, thanked us for coming, and just a very interesting man. Talked with him for, I would say, half hour perhaps, and then walked back out. Went back to the conference room and continued to work on the diagnosis. Excuse me. At the end of 3 days, we sort of summarized our findings, and I have to say that I don't think he had babesiosis. They had a blood smear where you can see the organisms, and sometimes debris occurs on those slides. And so I didn't see anything that I thought was truly Babesia. I made some recommendations on further tests they could do, such as a PCR, which measures the DNA of the pathogen. But couldn't say direct, I could not confirm that there was babesiosis. The other physicians thought, and I as well thought, that tumor was a possibility. And, we made some recommendations and hopped back on the plane and went home. We sort of joked, of course, we wanted him to get well, but we also joked that we prayed that he would not die within 3 months. Otherwise, we'd be counted accountable for that. So, or not making the diagnosis. But fortunately, he lived for a number of years. But anyway, that was sort of one of the high points I'd have to say of my career. It was really quite an event. So let me go from there to talking a little bit about how I wind up doing this research. I think it really started with Sam Matthews at Williams. I did a summer research project with him, and it really allowed me to understand much better how research worked and to realize that's something that I would be interested in doing in the future.
Peter, could you remind us, Sam Matthews, professor of biology?
Yeah, I'm sorry. He was the head of biology department at Williams when we were there. Yeah.
Thanks.
So, yeah. So, anyway, just talking also about the Williams family, I do want to acknowledge Bob Barr because Bob was the first to suggest that I go into medicine. It wasn't my parents. It wasn't, it was Bob. So that was good, and I took that advice. So as mentioned, I went to Tufts Medical School, which I really liked very much, and then took an internship and residency at Yale. And Bill Taylor also applied and was accepted. And so we were fellow interns, fellow residents. And Bill is just really a role model as a physician, as a house officer. It was really a good thing that it was really good to work with him. Um, I then subsequently went to UCLA. Jim Cherry was a renowned researcher in pediatric infectious disease, and he had a textbook that was sort of the definitive text. It was about two volumes, 3,000 pages. So he was known far and wide for that. Um, anyway, I completed 2.5 years there, then moved back east. Had hoped to get a job at Yale, but there were no openings. But there was an opening at UConn, and so I started there. About 2 years after I had started working at UConn, I get a call from Jim Cherry, asking me if I would write a chapter in his book on babesiosis. And I just thanked him profusely. I said, what an honor. It's just, I really appreciate, of course, I'll do this. And hung the phone up and said, what the hell is babesiosis? Uh, so I really had very little idea about what it was and started to read about it. Realized that if I'm going, I figured that book would go through many editions, which it has. It's currently on its 10th edition. I realized I'd have to have some street credibility and do some research in the field. So I looked through the literature and found that there was a man named Andy Spielman, who was a professor at the Harvard School of Public Health. He was the one who had done sort of more work than anyone else on human babesiosis. So I called him and asked if we could work together, and he said yes. And the first project went really well, and I worked with Andy. We wrote many papers until his death a few years ago. Um, Another person was Steve Weichel at UConn, who was also an entomologist like Andy, and then Derlin Fish, who is the person, entomologist at Yale, and really helped me get the job when I transferred. The final people that I wanted to thank are Gordon and Laura Gunn. They were Gordon Gunn's a billionaire. He once owned the Cleveland Cavaliers and brought LeBron James to the Cavaliers. But they have a house on Nantucket, and one of the family members developed babesiosis. There's a lot of babesiosis on Nantucket. So his physician, Gordon's physician, was Tim Leppery, who was a year ahead of me at Tufts, and I gave, just really talked through the case. I gave some advice on, how to handle the case. Unfortunately, the family member got well and all was good. About 2 or 3 months later, I get a call from Mrs. Gund. She expresses her thanks and says they'd like to support my research. And I said, not necessary, happy to just to help. She said, no, we really want to support your research. So I get a check in the mail about a month or two later for $300,000, and that was the first. I've gotten that kind of money yearly from them for the past 20 years, and I have 5 more years to go. So yeah, I really, am indebted to them, and so that's the story. That's how I got into it. So I'm happy to now just start the talk, and Alan, if you can advance slides for me, I really appreciate it.
Okay, here we go.
Okay, yeah. So we'll talk about Lyme and babesiosis. Next slide. And the outline is shown here. If you just, yeah, how I became, we already done that. Next. Overview of ticks and tick-borne disease in the United States. Next. Lyme disease, and then babesiosis, and then we'll finish with talking about prevention of tick-borne diseases. Have a slide on that. I think that's important. So next slide, please. Okay, these are the most common ticks in the United States. There are about 100 species of ticks worldwide, and there are about 10 species that are found in the U.S. These are the most common and the most problematic. And the very first one is click if you would again. The first one is Ixodes scapularis, which is also known as the black-legged tick or the deer tick, and it transmits Lyme disease, anaplasmosis, babesiosis, Borrelia miyamotoi, Ehrlichia murus, and Powassan infection. And click again, please. Dermacentor variabilis is the American dog tick, or also known as the wood tick, and it transmits Rocky Mountain spotted fever and tularemia. Next click. Rhiphencephalus sanguineus is the brown dog tick. It transmits Rocky Mountain spotted fever. Next. And Amblyomma americanum, the Lone Star tick, which causes human monocytic ehrlichiosis, heartland virus, tularemia stari, and alpha-gal meat allergy. The last item is really interesting. It's come to the fore in the past few years, and it's really problematic. And I at the end of the talk, there's some links. I've given this talk or talked about it with some newscasters on ABC, and you can click on the link and learn about alpha-gal. But if there's time, which there probably won't be, I'm happy to just speak on that briefly. Next, please. This slide shows Ixodes. Ixodes ticks are hard-bodied ticks. They're also soft-bodied ticks, but hard-bodied ticks are found in the northern hemisphere temperate zone. So if you look, you see in the northeastern United States and the northern Midwest, there's Ixodes scapularis, and it's the Northeast and the Northern Midwest are the two highly endemic areas in the world, actually, for Lyme disease and babesiosis. The tick is also found in the South and the Southern Midwest, but for reasons that are not fully understood, there really isn't much Lyme disease or babesia there. There's a little bit, but very little. Ixodes pacificus on the West Coast, Washington State, Oregon, and Northern California. In Europe, it's Ixodes ricinus, and then across Asia, Ixodes perslocatus and Ixodes ovatus. So these are areas where Lyme disease and babesiosis are found. Next slide. Just to give you some idea of the relative frequency, Lyme disease of these diseases, Lyme disease is clearly the most important. 34 in the CDC gathers reports, does surveillance every year. And in 2019, the last time they've sort of shown this compilation of all of these, 34,000 cases in 2019 of Lyme disease, 5,000 of anaplasmosis, 2,000 of babesiosis. Poisson virus, which is a very nasty infection causing brain damage in about half the cases. Fortunately, very small numbers, but they are increasing, and if this will really change the landscape if it turns out this becomes as frequent as these others. I will say the actual number of cases is probably tenfold what you see here. Surveillance is the best that can be done. We have a very strong public health system in this country, but there's still there are constraints in terms of being able to count all cases. So there's actually a lot more cases than what is shown here. Next, please. Next slide. Thanks. Okay, there's the Ixodes scapularis tick has 3 life stages. Eggs hatch in the larvae in the springtime, and the larvae are quite small. They're about, you take a pencil and make a point on a piece of paper, that's about the size of a larval tick. And larvae then feed on usually a mouse or other small rodents. They take a blood meal, which allows them to molt to the next stage. The next stage is the nymphal stage, which comes out in the following spring. And that tick is the one that's most responsible for transmission of Lyme and Babesia, that stage. That stage normally also takes a blood meal on a mouse or a small mammal, and that then molts to the adult male and female stages. They take a blood meal, and then the cycle is repeated. And I'll show you the life cycle shortly. Next slide. This is a slide. The tick on the far right is a female, is a male. Then you see a hat pin for size. You see then an adult female, and then finally an engorged tick. So they take a lot of blood when they take the blood meal. Next. This is the Paramiscus leucopus mouse, this white-footed mouse. This is the reservoir for these diseases. Mice carry one or sometimes multiple diseases that we've talked about. They have been living with these organisms for millions of years, and the mice do not become ill with them. They simply act as a reservoir for the organism. And when the ticks feed on infected mice, they become infected. Next. This is a tick tick bite. Normally, this looks quite, this is a person who's probably been bitten a number of times with increased inflammation that occurs around the bite. But you can see they are embedded, and they feed over 2-3 days, sometimes a little longer. They're hard to pull out, actually, unless they're fully engorged and they drop off. Next. This is a cartoon showing a tick taking a blood meal. If you look at the gray area to the right, you see these. You see sort of three prongs. The 2 outer ones are what are called the palps, and you can see them in the cartoon of the tick. They sort of stabilize the tick when it feeds. The central hypostome is like a syringe, and it has jagged edges. What the tick does is forces that hypostome into the skin and rounds around and makes a little capillary pool, or I should say a little pool of blood that it sucks up, and the blood goes into the midgut. And when you see the midgut in the tick, and if there are organisms there, they will get into the midgut, and that's what happens when the first when you have the first stage, the larval stage, takes the blood meal. Organisms, if the mouse is infected with Lyme, for example, Lyme disease, the organisms get into the midgut. Then with the second stage, when the nymph feeds, blood rushes in and the organisms move from the midgut to the salivary glands, those two green structures on the right and left of the tick. And what those glands, the organisms take a sort of a free ride into the skin, but the tick is actually spitting protein into various proteins into the skin, cement material to stabilize it, hemostasis factors that prevent this from clotting, so they have a steady flow of blood. They also have kinases like the breakdown bradykinin, which causes itch. So you're not going, they sort of prevent itch because they want to take their full meal and not get discovered and pulled off. And finally, they have inhibitory factors for the host inflammatory reaction, the immune reaction, because your body's going to react to these tick proteins. And over time, your body if you're bitten multiple times by the tick, you're going to have an increased reaction because the immune system gets better and better or stronger and stronger with each subsequent tick bite. So, for example, my entomologic friends who go out into the field and collect ticks have been bitten many times, and they know immediately if they're bitten by a tick because they have an itch and they can pull it off before these various pathogens are transmitted. Next. And yeah, so this is the life cycle. In the spring, the female tick lays about 2,000 to 3,000 eggs. These hatch into larvae. The larvae take a blood meal from a mouse, and if that mouse is infected with any one of these, Lyme, Babesia, Anaplasma, any of these pathogens, the ticks then pick this up. They then overwinter, and the following spring they molt into the nymphal stage because of that earlier blood meal. And then if they then in turn bite an uninfected mouse or human, they become infected. After that blood meal, usually on a mouse, or again, small mammal, they molt into the adult stage. The adult male and female prefer to feed on deer. They can feed on other mammals, but they much prefer deer. And on the deer, they procreate. And they also, the female gets enough protein from that blood meal to lay her eggs the following spring. But deer can carry hundreds of these ticks. And wherever deer are, the number of ticks is greatly multiplied. And that's important in understanding that the epidemiology of the disease, which is covered on the next slide. This slide shows Lyme disease emergence in the United States from 1993 to 97 in the dark red, 98 to 2002, 2003 to 2007, 2008 to 2012. The original Babesiosis was discovered first in 1968 on Nantucket Island. It's actually the second case. The first was in California, but they have very little disease there. But Nantucket Island is where the second case in the United States was discovered. And this map begins in 93. So we're talking a number of years after the introduction of Babesia on Nantucket, and also a number of years. The Lyme disease was discovered in 1978, I think was the first paper. And that was in Old Lyme, Connecticut, which is near New Haven. And from that site in New Haven, or near New Haven, I should say, in Old Lyme, the disease is spread, and as you can see, it's gone north, south, and west. And by 2012, it was extending from Maine down to Virginia. Likewise, in the Midwest, the Minnesota and Wisconsin were independently infected or had infected ticks, and they have moved. Both are moving toward each other in the Michigan area. So why was there this expansion? And the hypothesis is that when the colonists arrived, they started cutting down. This was all by the 1900s. There was very little forested area in the Northeast. There were these refugia of deer, deer herds. There are just a few deer left because their habitat had been destroyed. There were so few. There was a headline in a New Hampshire newspaper about a deer sighting around 1900, early 1900s. But then what happened is agriculture moved to the Midwest, the forest returned, and with that, the deer returned. And with that, the number of ticks and tick-borne diseases started to climb. And so we've seen this emergence over time, based on probably on the deer and how it fits into the life cycle of the organisms, of the tick specifically and the ticks. So, okay, next slide, please. This shows that Lyme disease is really, again, the number one cause of tick-borne disease in the United States. And it has a very broad geographic range. And that's the dark gray. This shows the human cases reported between 2011 and 2013 to the CDC. The dark gray areas are areas where there's both Lyme and Babesia. The green areas are where there's just Lyme, and there are no areas where there's just Babesia. So essentially, Babesia is not transmitted as readily, and it's sort of moving. It sort of follows Lyme disease geographically for reasons that we don't fully understand. But what that translates into is far fewer cases of Babesia. But nonetheless, it started out as a rare disease. When I first started working on the disease, there were very few cases, and then subsequently it's been recognized that it is worldwide and it is a major health problem. Next slide, please. This shows the number of cases of Lyme disease from 1996 through 2012, and you can see that there's a steady increase. There was a drop off in 2019, 2020, 2021. That was attributed to the fact that that was when COVID hit, and the CDC resources the surveillance resources really were cut back for everything except COVID. And so this is probably not a biological event. This is probably has to do with just funding. By 2022, the funding had returned, COVID was under control, and they actually had a new case definition, which is a little more liberal than the previous one. So here we are at 60,000 cases. And again, it's estimated 400,000 to 500,000 cases a year for Lyme disease. Next slide. The same thing is true for Babesia. There's been a steady increase in the number of cases. Again, much fewer than Lyme, many fewer than Lyme, but between 2,000 and 3,000. And the actual numbers probably exceeds 30,000 would be our best guess, but we don't know that for sure at this point. Next slide. Okay, let's talk. I just want to talk about these two specific diseases then, a little detail, and then we'll be done. So the causative pathogen of Lyme diseases are Borrelia species. There are a number of these are bacteria, coiled-shaped bacteria. I'll show you a picture of one coming up. Next slide. Or next. Transmission is by, again, hard-bodied ticks. Next. The geographic distribution is worldwide. Next. Target tissue is skin, joint, cardiac, and neurologic tissue. Next. Clinical manifestations are usually a rash called the erythema migrans rash, which is a red expanding rash. I'll show you pictures of that shortly. Or you can have a viral-like illness. Generally, about 80%, 90% of the time, people do have an EM rash, but about 10 or 20% will just get a viral-like illness, which is they'll have fever, they have joint pain, they have muscle pain, fatigue, headache, and so on. The diagnosis is with an epidemiologic medical history. The diagnosis, if someone's living in Arizona, the chance that they have Lyme disease, if you see a rash on them, is very low. On the other hand, Connecticut, Massachusetts, New Jersey, wherever you're going to, in endemic areas, it certainly should be on your diagnostic list, especially when this occurs in the summertime. Physical exam is key because the erythema migrans rash is the way the disease is usually diagnosed. You can do some lab tests, antibody and PCR. PCR preliminary chain reaction to look for the DNA of the organism, but those two are not nearly as useful really as the EM rash. Next. Treatment is with doxycycline, amoxicillin, cefuroxime, all given by mouth. And azithromycin can be given IV or by mouth. Next. Also, I shouldn't point out ceftriaxone is another agent that's used. Borrelia burgdorferi pathogen is shown here. This electron micrograph on the right. On the left, you can see they're a coiled organism. They coil and uncoil, and that propulse This is through tissue. So they move around a lot. This is from a tissue culture dish. You see the organisms are photographed there. Next. Clinical manifestations. There's the early localized stage. Next. That's with the erythema migrans rash at the tick bite site. Again, 80–90% of people get that. Or a viral-like symptom, 10 to 20%. Early disseminated stage, it disseminates out from where the organism was originally placed. Next. So you can get multiple erythema migrans about 10% of the time of all the cases. Cardiac involvement about 5%. Neurologic manifestations, Bell's palsy, which is a facial paralysis, usually transient but sometimes permanent. And then meningitis in about 10% of cases. Next. The mortality, fortunately, is quite low. It's less than 1%. Next. Late stage disease is arthritis. And when the disease was first discovered, what researchers noted was that people would get the erythema migrans rash in the summertime, and then in the fall, several months later, they develop arthritis. The knee would swell. It's usually the knees that are involved. They become swollen, a little bit painful. And it occurs in about 30 to 40% of people that are untreated, whereas if you're treated, less than 10% will develop arthritis. Next. Chronic neurologic conditions in 5%, and those conditions are shown next. Shooting pains, numbness, tingling of the hands or feet, problems with short-term memory. Next. There's an entity called post-treatment Lyme disease syndrome. That is, some patients, despite standard therapy, will have persistent symptoms. This occurs, it's hard to know exactly what the figure is, but it's probably somewhere around 10%. Next. These people have muscle and joint pains, sometimes cognitive defects, sleep disturbances, and fatigue. I've certainly encountered this with some pediatric patients, but also adult patients. I get consulted on them as well. And it's really unfortunate. It's really a problem. We don't understand why this occurs, although people are doing research to understand this. And it really causes long-term problems for folks. Next. This is an erythema migrans rash. As you can see, it's a large oval. This picture was taken. This is an early photograph of a Lyme disease patient. It's before we knew what the cause was, before we knew how to treat. People would get this rash and would extend. I mean, this is 5 inches in diameter. You can see that there's an outer red ring, and that's because the organisms start at the center of that oval, and they're injected. They cause redness there, and the redness moves out as the organisms are moving out. And when they first hit the immune response of the host, you get the darkest red area. So this is, sometimes you get a red circle in the middle, and it's called the target lesion. If the lesion is 2.5 inches or greater in diameter, that is 5 centimeters in diameter, and it's an expanding red rash in the summertime, most physicians are going to make that diagnosis of EM rash and treat the patient. Next. This is a child who has disseminated disease, and you can see the lesion on the left is the original lesion. Then you can see these other lesions, one on the right, one above the original lesion, and then a few on the right. So the organism is spread either through the bloodstream or the lymphatic channels to other parts of the skin, but you can also get dissemination to the brain, the heart, or the joints, as I mentioned. Next. This is not all the rashes are the bullseye rash. You can get we call a confluent rash, all red. Person was bitten in the arm, in the axilla, and that's another EM rash. Next. Clinical manifestation timeline. Next. Actually, you know what? I'll go through this really quickly. Time of transmission, from the time the tick bites to transmission of the organism is 36 to 72 hours. Incubation period is 1 to 4 weeks after the tick bite. So you can go a month after you've been bitten and then develop symptoms. The duration of symptoms, usually 1 to 2 weeks. Next. Sometimes years, though, as we've talked about. Lyme disease vaccine. Next, sorry. Lyme disease vaccine. There was a Lyme vaccine that was developed around year 2000. It was 80% effective, but it required 3 shots and yearly boosters. And so it was not really, not a lot of people decided to take the vaccine. And then there was a lawsuit that people would develop arthritis after they got the vaccine. However, that turned out to be a red herring or a false alarm, because in the clinical trials, they compared around 10,000 people with the vaccine and then those without. The amount of arthritis in the two groups was the same. But nonetheless, because of this class action suit and the poor sales, the vaccine was discontinued. Next. A European company, Valneva, has developed a new vaccine. Next. Valneva joined with Pfizer to do a clinical trial comparing the vaccine with no vaccine. Next. Phase 1 and 2 trials are completed. Those really look largely at safety to make sure the vaccine's safe in a smaller number of people. Then you look at efficacy in the phase 3 trial, and phase 3 trials are now being carried out. They've been delayed, but we should have the vaccine in another 2 to 3 years. Next. Next. Okay, babesiosis. Go ahead. Causative pathogen or Babesia species. They are protozoan parasites. Next. Most common are Babesia species infecting human. The most common species is Babesia microti. That's what we have in the United States. Next. Target tissue are red blood cells. These organisms get into red blood cells. They're found in the northern hemisphere, as we saw that picture. Transmission is basically tick, but you also get it through blood transfusion. Because when you get this infection, for about 20% of adults will be asymptomatic. They don't realize they have Babesia infection. They go to give blood. The blood is given to people who receive blood are often immunocompromised, so the recipient is often quite ill. The mortality rate in that group is 20%. An infected pregnant woman can transmit it to her child. That's pretty rare, but it occurs, and it also occurs with organ transplantation. The diagnosis here is, again, by entomology, do you live in an endemic area? You get a medical history to see if the symptoms are compatible. Physical exam, usually you can sometimes feel enlarged spleen or liver, but we really rely on the blood smear, PCR, and antibody, but especially blood smear and PCR to make the diagnosis. Next. Treatment is with a combination of two antibiotics, atovaquone, azithromycin, or clindamycin quinine. Next. This is a slide showing the organisms, and you can see these are red blood cells, and the ones that are clear are uninfected, but you can see those little circular dark purple structures. Those are Babesia parasites, and this is what when I looked at the slides in China, they did not reveal something quite this classic. So there was questions, but anyway, this is the main way in which the diagnosis is made. You can also, as I say, do a PCR on blood, which amplifies the DNA and tells you that the disease is present. Next.
Peter, what's a PCR?
I'm sorry, that's short for polymerase chain reaction. And essentially what you do there is the DNA of Babesia are certainly different than the DNA of humans. So you can amplify the DNA from a small number to a very large number, and then detect that with through the methodology. You can tell that the DNA comes from the Babesia, not from, and so if the PCR is negative, there's no Babesia DNA there and there's no Babesia. But if it's positive, it tells you that the organism is present. Clinical manifestations, asymptomatic infection, 20% of adults, 40% of children. The typical illness is a viral-like illness with fever, chills, sweats, headache, fatigue, and malaise, anorexia, nausea, cough, muscle ache, and joint ache. Severe illness involves the heart, the lung, the liver, or kidney. All may fail, or any one of those. And you can get a persistent relapsing illness despite standard therapy. These are in people who are highly immunocompromised, and that can go on for months or years. Next. Death is anywhere from 1% in healthy individuals to up to 20% in some of these folks who are immunocompromised. Who's the at-risk populations? People over 50, like you and me. We would get more severe disease than the average person, but we wouldn't get it. It would not be as severe as someone who has no spleen, asplenics, HIV/AIDS, malignancy, or get it through blood transfusion. Timeline. It's like Lyme. It takes 36 to 72 hours for the organism to be transmitted once the tick bites. Incubation period, you can be well for up to 6 weeks and then develop symptoms. And if you have had a transfusion, you can be well for up to 6 months and then develop symptoms. Duration is, again, usually a week or two in people who are immunocompetent. Next. Sometimes more than a year in highly immunocompromised hosts. Next. Okay, this is the last slide, everyone. I want to talk about prevention. So click down again, please. Personal protective measures. Okay, avoiding tick habitat. Ticks live in the woods generally. And so if you're in the wood or tall grassy areas, you want to avoid those areas if you can because they're more likely to, that's where the deer are, that's where the ticks are. And the mice are there too. Um, if you have to go in such an area, it's good to wear long sleeve shirt, long pants, and to pull your socks up over your pants. Not very pleasant to do in the summertime, but if you want to be safer, that's what you do. You can also put on DEET or on your skin or permethrin on your clothing. These are acaricides. They kill the ticks. Next, tick bite prophylaxis. Next, tick checks. So after you come in from a wooded area or an area where you think there may be ticks, you can do a tick check in the front. You need a partner to check your back. You need to look in the hairline also because it's sometimes missed because of that. And there's a way, there's been research to show that if the tick has been on for less than 36 hours, you're not going to get Lyme or Babesia because it takes that long to transmit. But it turns out if a tick has been on longer for that, if it's been on for, up 36 to 72 hours, you can take doxycycline and that if Lyme was transmitted, it will kill the organism and you will not get Lyme disease. Um, if the tick's been on for more than 72 hours, then doxy is probably not going to work. Next, landscape modifications. Okay, you can cut your grass, keep your grass relatively low, and leaves are especially problematic. Leaves retain moisture. Ticks tend to dry out in the summer, and so if they crawl under a leaf, they get shade, they get some moisture, and that. So you want to clean up leaf litter on your lawn. Next, chemical control. You see this gentleman spraying. These are synthetic insecticide or natural compounds, and it's effective, but you need to do it each year. Next. Okay, let me just mention the top picture, which isn't mentioned here in the list. It's a barrier of stone where you can put wood chips between your property and the lawn. Ticks don't like to go across that. That may afford some protection, but an entomologist friend of mine who's studying this said it's not terribly effective. Spraying would be more effective. Host targeted caricides next. So I mentioned DEET and permethrin for humans. For deer, there's a thing called a four-poster device where you put down grain in an area and you have four posters, kind of like a paintbrush, or they put acaricides on these posters, on these posts. And as the deer go in to feed, they rub against this acaricide and it covers their body, and it really has been shown to work. The problem is it's expensive. Rats and mice like to congregate there, so it's not really widely used. And there's a little mouse house that's been created where they put peanut butter in a little plastic house. The mouse goes in, there are rollers there that apply the caricide, and that has not been shown to be effective, but people are still looking at that. Deer reduction has been done on islands, and there's several examples of deer have been eliminated on those islands. The number of ticks has dropped dramatically, as has the cases of tick-borne infection. Um, and you can exclude them by using a high fence. Deer can leap about 6 to 8 feet, but an 8 or 9 foot fence pretty much will keep the deer out of your property if you're interested, if you want to decrease your risk. Next. Host-targeted vaccines. So we talked about the Lyme vaccine. Next. Finally, education and behavioral change. If you know if you understand what we've just talked about and do further reading, it can help to prevent disease, obviously. Okay, I think so these are the media links, which I think Alan or John already sent out. I did a little talk with ABC on babesiosis and on the alpha-gal syndrome. And I think that's it. The last slide is a picture of Block Island, where been doing studies there for about 30 years. There are about 1,000 people on the island, about 1,000 deer, and many more ticks. But it's lovely. It's a beautiful, beautiful island. And I thank you very much for bearing with me. I'm so sorry about the computer problems, but it is what it is, and here we are.
Thank you, Peter. That was really terrific. One of the things that I remember going back to our 50th when we had sat down and had a couple of drinks and talked about Lyme's disease, was the sense of the carryover some people after the disease has left them, having the symptoms that they had during their Lyme's disease. One of the things I found was short-term memory was greatly affected by Lyme's disease. But one of the questions that I had for you was that you've been working on Block Island, and you've been trying to interrupt the cycle in some way, shape, or form. No success at that, really?
Yeah, very good question. So we were I mentioned Andy Spielman. He was invited out to Block Island. There was an MIT professor out there. They had a group of people that knew that there were huge problems on Block Island, as there are on most of the islands off the coast of New England. And they wanted to eliminate the deer on the island because Andy had already done this project where he had gotten rid of deer on another island off the coast of Massachusetts, Great Island, and it was very successful. So what their real objective, they invited Andy to come to the island to define what the problem was. They already knew what the problem was. They had a lot of Lyme disease, a lot of Babesia. Um, that was the overt reason. The covert reason was to get rid of the deer, and hoping that we would, in doing our research, would point out the very high rates of disease and argue and show that if you got rid of the deer, you could probably reduce the risk. So we went out there and started working. We worked with a physician there. We did what we call serosurveys twice a year. It was very successful in terms of better understanding how the disease worked. We tried to be apolitical, not take sides, because there are people there that have very strong, everywhere have strong. Some people dress up in deer costume during hunting season. Will go out there to prevent hunters from doing that. So, but the bottom line is we never got to the stage of reducing deer. It was just too politically charged.
So that didn't work. Anybody else have some questions? Ken.
Well, Peter, great presentation. Thank you. The question I've got, you've got all these mice running around with Lyme disease and babesiosis or whatever. Can you get it just from them directly, or do you need a tick intermediary?
That's an interesting question. I think it's theoretically possible to transmit the disease. If you were skinning a mouse and you had a cut on your hand, it's theoretically possible you could get Babesia or even Lyme disease that way. I think it's unlikely. I mean, it's never been reported. So I think the practical answer is no. Ken, I have to say one more thing. I was going to say this, but I decided maybe not to. You'll find out after I say it. But you and I took an embryology class. I was a bio major. You were not a bio major. We both did well in the course, but you did a little better than I did. So I thought maybe I should thank you for not applying to medical.
My pleasure.
Okay.
Ed Helm.
Yeah, Peter. So there was a friend that was going to link into this because she got a tick disease that's a new tick disease. And she lives and is an artist in Lexington, Virginia. And suddenly she got sick and she had some meat at a nice restaurant in New York City and she started throwing up. And nobody could figure out, or doctors couldn't figure out what was wrong with her. One of her friends said, well, have you been bitten by a tick recently? And she said, yes, as a matter of fact, I had. And apparently about, a week before this, she was bitten and around her crotch or in a private area, but, definitely she was bitten. So is this a brand new, what is this disease that seems to be unknown to most of us that think about ticks, but we should be having that on our radar? Have you listed that?
Yeah, that's a great segue into it's the alpha-gal meat allergy problem. And essentially when, our immune system reacts to any foreign body or any foreign protein and certainly both cancer as well, which is abnormal tissue, but also to pathogens. And we eat red meat all the time, and we have no trouble with it, even though we're ingesting, in a sense, this foreign protein. The immune system is remarkable, and certainly there's a very elaborate immune system in the gut that prevents us from developing an allergy when we eat food. Although people do have food allergies, it's not 100%. You can get peanut allergies, whatever. So what happened? What people discovered was this. They basically found out that the ticks, and especially the Lone Star tick, if you're bitten by that tick, ticks feed on mice, and they pick up this glucose, this sugar moiety, alpha galactose, and they pick that up, and when it's injected in the skin, all of a sudden, if you take red meat, if you when you eat red meat, you're going to develop really severe, or you can develop very severe reaction, GI reaction. I mean, it ranges. It ranges from slight stomach upset to life-threatening anaphylaxis, and so it's really quite disturbing. And the you really can't eat red meat, and you have to wait sometimes years before you have it again. And you have to avoid tick bites. So people are working on a vaccine for it. The tick, as I say, most responsible is the Lone Star tick. That is a tick that's been found in the South for many years, but it's moving north because of global warming. And now it's found, apparently on Martha's Vineyard, for example, there's a lot of it. But I think there's a lot of it, in general, in states that never had it before. I think the research on this was done about 15, 20 years ago. So it's relatively new and certainly a growing problem. But if you go to that link, the really, you can, I speak about it, and or you can certainly look it up and on the computer and check it out. But yeah, I feel badly for her. But there are things that there are some medical approaches that you can take to ameliorate it. But yeah,
Thanks, Doc.
no
Bill
problem.
Clendaniel.
I had a professional on the other side of the coin, so to speak, when I was managing properties for the trustees of reservations. Carol Perry may remember this, but we had a major deer problem at Crane Beach, which was a 1,300-acre property that we owned on the coast of Massachusetts, very popular swimming beach. And it was being overrun by deer eating the vegetation on the barrier beach, which was threatening the whole property. And as the owners, we were very anxious to have a deer hunt. And that raised all the hackles that you mentioned, Peter. And I got to work with Andy Spielman. It's nice to hear about him.
Oh, really?
Again, this was in the mid 80s.
Mhm.
And basically, I mean, we really had all out war with the people who wanted to not kill the deer, and then the people who did want to kill the deer. And it was actually Lyme disease and the tick that saved the day, because people started getting Lyme disease, and that was the weapon we needed to get the deer under control. So helps.
Interesting. Yeah, I can say that clearing deer from the islands is relatively easy. I mean, it's not easy, but it's relatively easy compared to the mainland, would be a tremendous undertaking. I mentioned Powassan virus, and I mentioned that it's about a 10% mortality, and about half the patients have permanent cognitive, disability. I think if Powassan becomes very prominent, very frequent, if that continues to increase, I think the deer will probably be history. People will just not put up with the idea that they have a fair chance of permanent brain damage as a result. These other diseases can do some nasty things, but that would really, I think, probably change change the landscape. But yeah, it's very interesting how the whole deer management issue. One final thing I'll say is it's possible. Some research suggests that you don't have to get rid of the deer, but at least cull the herd down to 10% or so, and that might be effective.
We're talking about what remedies.
John Way, you've got a question. John Way, did you have a question? Oh, you got.
You can talk. John, you're on mute.
Okay.
I've heard that the animal that has um, a hairless tail. I'm blocking the name of the animal. Anyway, it's said that this animal eats, thousands of ticks. Is there other? Are there other? Is this possible that it's a marsupial animal?
Yeah, I don't, it's funny. I think I vaguely remember that, but what I really remember are guinea hens. Um, and who was married to Billy Joel? She was a model. Anyway, she had guinea hens. It was
Opossum. Opossum. That's what it is.
okay, good. Yeah, I mean, guinea hens or even just regular chickens have amazing eyesight and they eat ticks. And but and some people feel, I mean, let me say this, there's never been a study that I'm aware of that shows that ownership of guinea hens or chickens in their backyard has decreased their risk for Lyme disease. Um, it's the numbers are such that probably not, but who knows, Brad? I mean, people have used that for that very purpose. Um, so, yeah, it's interesting. Never seen
There
it.
was one other story I wanted to tell. In 1984, when I got married, we went out to Missouri, and we went to this park, and the park was virtually empty. And they provided a little cabin we could sleep in. And anyway, we got back from the walk. We looked down at our legs, and they were, it seemed like hundreds of ticks climbing up our legs.
Yeah.
And so we never had any bad effects from those ticks. But I just wondered, did Lyme disease ever appear in Missouri over the other side of the Mississippi River?
I believe Missouri has had Lyme, but also there's a disease called STARI, Southern Tick, forgotten the acronym. Anyway, there's a Lyme-like syndrome in Missouri, but um, I think they've had a few cases, but not many, not like Wisconsin or Minnesota. Um,
We had to realize that everyone else seemed to know about the ticks except us because
you
we
know,
were the only ones in the park.
it sounds to me like you may have stepped in an Ixodes murus nest. These are the Ixodes scapularis ticks are real pioneers. They go out far and wide. They range, and they're very aggressive ticks. The Imurus ticks don't travel far from where they're wherever their host is. So if it's an opossum or maybe a chipmunk or where they stay in that area, just around the nest. But there are a lot of ticks in that nest. So I suspect you may have stepped into a nest there, and that's
Yeah.
what you did.
Thanks, John. Ed,
I live in a very tick plentiful area. I mean, I'm in Poughkeepsie, and but I have found one thing: if you have clothes that has repellent in it,
Yeah.
it will vastly decrease the number of ticks you're likely to get bitten by. And I learned this when my brother and I went walking in the woods, and he had those pants, and I didn't, and I got 5 or 6 bites, and he got none.
Yeah. Right.
work.
Right.
Thank you, John. Ed Wing, you've got.
Yeah, so Peter, that was a great summary,
Thank you,
and
Ed.
I really appreciate that.
Yeah.
I'm living in Jamestown, Peter, so
Beautiful.
another island off of the New England coast, and it's
Yeah.
like Block Island, the incidence of the disease. But I wanted to comment for the group that Peter is somebody who infectious disease people and other physicians in the academic communities, particularly in New England and around the country, will call with difficult cases of Lyme disease or babesiosis. He's the go-to guy in the country in a way about these diseases. So it's a huge
Thanks, Ed.
credit to you, Peter, but a very
But
nice.
I'll tell you a little story about that. As I say, when I started, there was very little disease. So I would say to people, you've heard of a big fish in a small pond. I would say to people, I'm a big fish in a puddle. But Yeah, I mean, it's just because it took a while and because the NIH did not, I mean, I had a few NIH grants, but it doesn't fund that very well. It's sort of like, there isn't a lot of competition because everyone is studying Lyme disease or, anaplasma or whatever. But yeah, so yeah,
But
thanks.
Block Island was a perfect laboratory, right?
It was just the perfect place. And
Yeah.
because we could work with a physician there and do this sero surveys, which captures asymptomatic infection, because you can get a person who's negative in the spring and positive in the fall, and if they've had no symptoms. So we were able to calculate asymptomatic infection rates. So it was really cool.
Yeah, yeah.
That's great.
Great.
Alan,
Yeah, Peter, I have a question. If my cat catches a mouse and plays with it in its mouth for 10 minutes, I'm in New Hampshire, as you know. Do I need to be concerned?
I don't think so. Uh, I've never heard of a case where sort of blood from an animal is going to, unless you have a cut on your hands. But again, it's never been reported as far as I know. But you do bring up the possible, what's the story with pets, with cats or dogs?
Yeah.
And it turns out that your risk of tick bite is increased if you have a dog or a cat, but no one's ever shown that the incidence of Lyme or these other tick-borne infections is greater if you have pet ownership, and that's because only about something on the order of 25 to 30% of ticks are carrying Lyme disease in highly endemic areas, like in Connecticut or Massachusetts, whatever, New York. Only about 25% or so are carrying the organism. And it's not always transmitted. If an infected tick bites you, it won't always transmit the disease. So I think there have been prophylactic studies looking at doxycycline. They found that maybe of all tick bites, something on the order of 3-4% go on and develop the disease. So the chance for, unless you're bringing your pet in every day and there are ticks all over you, your risk is still probably, low.
But Peter, we've had pets in northern Vermont, and we could take them to the vets, and they'd get a shot for ticks,
Yeah,
vaccine of some
Lyme
sort,
disease.
yeah.
and for Lyme disease. But they couldn't give it to us, or there was nothing for humans.
Right. Well, there was the human there was human vaccine back in the 2000 or early 2000 I mentioned. There was a human vaccine, but again, it was stopped. There's always been a dog vaccine, and some people have been so desperate they've taken the dog vaccine, which I would never do, but people have done that. Dogs get Lyme disease, and protecting them is important. I want to mention one of the really interesting things. Remember, I said that these ticks are spitting protein into you to prevent blood clotting and prevent itch. You could make a vaccine out of those proteins. And then if you gave a person a tick vaccine, not only would they not get Lyme disease, they wouldn't get Babesia, wouldn't get many of these infections. So it's a more generic effect. And it's very promising. People are working on it, not yet developed, not yet perfected, but they're doing that for both mosquitoes and ticks.
Can we talk about big pets?
Bob Bear.
Hi, Peter.
Hi, Bob.
The population of deer, I'm slightly south of Allen, but not very much in New Hampshire. And the population of deer in the last 5 years has probably quadrupled.
Really?
We have about a mile dirt road to the area where we live with, not alone, but with a whole bunch of people. And probably once every few weeks, I used to see a deer in the woods or crossing the road. Now I'll be driving down the road and there'll be 5 deer sitting there watching me completely. They used to run away as soon as they saw a car coming or a person walking by. Now they just stand there and wait for you to walk by.
Right.
So I have a feeling northern New Hampshire is going to have a burgeoning babesiosis or Lyme incidents coming up.
Yeah, they're still moving north, and global warming is, contributing to that. But yeah, the deer are really bad. They're very smart animals, and they probably talk to each other, and They say it's just a car, don't worry about it, or wait till whatever. No, they really are. They're really amazing creatures. But I don't know. I really think it's a little out of hand, and there should be, various places try. They'll have a hunt to
Yeah.
try to reduce the herd. But again, the data right now indicates unless you get down below 10%, it's probably not going to have much effect.
Thank you, Roger Walkie, and then Rob Jones. You have your hand up too. So, Roger, why don't you get?
Yeah, Peter, the map you had showed occurrence zones on the coast of California. And I was puzzled about what deer populations would be there.
That's a very good question. Okay, so I don't know California as I know the East Coast. There is a species of deer I think out there that probably support the tick population, but there are other mammals that can serve as hosts. The amount of Lyme disease and Babesia on the West Coast is much smaller than the East Coast. And there's a very interesting reason for that. It may be the hosts are not as plentiful, but one entomologist was able to show that it turns out that the ticks out there, they're lizards. The Western fence lizard is about 3 or 4 feet long, and ticks feed on the lizard, and the lizard has a complement system, which is part of the immune system that kills the organism. So it's sort of like if you have an infected larvae I'm sorry, if you have a larvae that gets infected, becomes a nymph, the nymph feeds. The nymph will cleanse the nymph so that the adults don't get it. It just decreases the amount of Lyme disease and these other pathogens. That is the fact that they're feeding on this Western fence lizard. And it also well, anyway, it's sort of like the whole sequence stops in that instance. But anyway, it's a good question about the deer. I vaguely remember a deer species out there that probably does contribute. But the number of babesiosis cases there've been I think less than 50 cases, and there's several thousand that are reported each year from the combined rest of the U.S.
Thank you, Rob. Did you have your? No. Okay. Any other questions? Because I think
Jack Hunt had his hand up. Jack, you need to unmute.
Unmute. Oop. There we go.
Interesting story with that OBS. How do you pronounce it? I never could get it pronounced right.
What?
It affects horses.
Okay. And what was the term? I'm sorry.
We call it obeosis. I don't know what.
Oh, I'm sorry, babesiosis.
Babesiosis.
Yeah. Sure.
When I was with King Ranch, we had a big horse sale on an anniversary, and we sold several very expensive quarter horses. And a few weeks after that, one of our using horses came down with Lyme, what would be a horse version of Lyme disease, babesiosis. And we had to. It's a reportable disease in terms of the USDA. And so we had to go through a lengthy process of not only recalling the horses that had been exposed to the horse that had the disease, but terminating the horse that had the disease and going through a huge protocol with USDA. I spent, I don't know, a couple of weeks off and on up at USDA in DC working on this problem because for King Ranch it was terribly embarrassing. And we think that the disease or the ticks that caused the disease may have come from Brazil, where one of the horses in that same herd had originally been in Brazil and they brought him back up to southern Texas, which is a very heavy deer area anyway. But it was an interesting and very expensive deal and very difficult public relations deal. So it's not trivial at all in terms of the practical effect in agriculture. So
absolutely. No, I mean, that's a very important point. As I mentioned, there are over 100, there are over 100 Babesia species, and they infect animals, both wild and domestic. And so horses are affected. It has been and is a major problem in cattle. cattle get several Babesia species. Divergence is the most important. And so ranchers play I don't know, you can tell me, but my understanding is, cattle are dipped. They go into this large vat where they get covered with the, acaricides. And I actually went down to McCallum, Texas with Andy Spielman many years ago because McCallum's like at the tip of Texas, and there's the Mexican government cannot afford, or in general, Mexico can't afford the measures that U.S. ranchers can to prevent tick infection or tick bites on their cattle. And so there's this sort of neutral zone, a 10-mile neutral zone that we learned about between Mexico and the U.S., between Texas. And anyway, that was very interesting visit. But cattle and horses are infected, and there was some racehorse that got infected. I think it was in the Kentucky Derby, and had to drop out because of babesiosis. But yeah, it's a major economic concern.
Thank you very much, Peter. This was really just a great presentation, and we really have enjoyed having you do this. And we just all want to thank you.
Thank you so much. And if you have any technical problems with computers, just give me a ring.
Well, we thought we had it all worked out, but it worked out.
I mean, it's all my end. It's all my end. In fact, this computer's on its last legs and I'm getting rid of it, but I haven't been able to do that yet. But anyway, I paid the price for waiting, I guess.
Well,
thank you very much.
The honorarium you got from us will help in terms of computer. But probably not. Ken, I don't know whether you wanted to say anything about, you would send out something to everybody about military services, one of the ones that we wanted to work on. And I don't know what kind of response you've gotten, and because this is going to be for sometime in the beginning of next year.
Yeah, what we were talking about was having a panel of classmates who had gone in the military in one way or another. I sent out an invitation. I've gotten maybe six responses, and which is fine. I mean, that would cover probably our time, but I'd welcome any others that would be interested in participating as well. And it's a whole spectrum. So far, I mean, of the responses I've gotten, amazingly different military experiences, whether, some people went to Vietnam, some people went to Germany, some people went to Australia. I mean, it's really a fascinating spectrum. So anyway, I think it'll be an interesting session. I think we're planning, as you said, John, early in the year, January or February, something like that. But I would invite anybody that did have a military experience that hasn't responded already to send me an email and kind of summarize what your experience was, and we'll try to slot you in. I think it'll be kind of interesting.
Thank you. That's great. And the other thing we talked about it earlier, but our trip to Montgomery, which really should be a tremendous trip. Alan had sent out through listserv, and actually, with Don Brown is really going to do a wonderful job for us. But Alan sent it out through listserv for response by November 15th. But anybody who wants to join us, we really would love to have you. I think it's going to be an excellent weekend, and we'll be learning a lot about what's happening still in Montgomery and what happened during the time when we were doing the civil rights work. And it's really going to be a very interesting time. Alan, I don't know whether you want to add anything to that.
I see Don Brown is here under the pseudonym of Ron Bodenson. Don, did you want to say anything?
Ron Bodenson.
Ron's name's been jumping around.
Well, he loaned me his Zoom piece. Listen, two things. I hope to give it to you in easy bites where you can understand again and refresh your minds about what happened between 67 or 63 to 67. Most of us were interested in the drive to Smith, Holyoke, Vassar, et cetera, and the philosophy paper due the next day. But I got some interesting people that you'd love to meet that will give you a fresh idea. It'll be a learning experience, and there's some fun places to see. And most of all, it'll be a good convivial time. And I see that Pete Krause is Peter. You're signed up, so I'll ask all my sneaky questions about Lyme disease when you're over a cocktail. And we look forward to seeing you. It's early June. We'll send out another notice to see if you can work it in. It'll be a fun time.
Well done.
So far, we've got 18 classmates who've said they're interested, and with spouses and partners, that's 33 folks. But 18 classmates is a good start. But we'd love to have as many of you as can make it. So that would be great. You could drop me an email, show that then Don will know how many rooms to reserve. And Peter, you did mention 2 links, which I'll send out as a follow-up once we get the recording of this up posted online.
Thanks a lot, Alan. Appreciate it. And I appreciate you and John helping me with this computer.
Okay. My pleasure.
Thank you, Peter. It was really a great presentation tonight. And thank you all for coming. We really love to have the full class together and as many people as come. And so we'll know we each look like. We'll have had some conversation, certainly for our reunion, which is coming up in 2 years. And we're going to get started on putting that all together. So looking forward to seeing you all there.
Okay. Have a great holiday, everybody.
Yep.
Same to you.
Hey.
Y'all. And thanks for
Right. Thanks.
Thanks.
Thanks.